The black tumors on these fish did not begin inside the fish carrying them. That is the conclusion hidden in their DNA. In Lake Memphremagog, a long lake crossing the border between Vermont and Quebec, biologists had been finding brown bullhead catfish marked by dark, raised lesions. Some looked like patches of ink. Others grew into heavy, crusted masses. At first, the likely suspects were familiar. Pollution. A virus. Something in the water that made many fish develop their own cancers. But when researchers compared healthy tissue with the tumors, the expected family tree collapsed. The healthy cells belonged to each individual fish. The tumor cells did not. Across different animals, the tumors shared hundreds of thousands of genetic variants. They were more closely related to one another than they were to the bodies in which they were growing. This was not simply cancer appearing again and again. It was one cancer lineage moving from fish to fish. For the first time, scientists had identified a naturally transmissible cancer in a fish and in a freshwater animal. But that discovery opened a darker question. If the tumor can leave one body and survive inside another, when does a cancer stop being only a disease and start living like a parasite? This is the cancer that escaped its original body. Brown bullheads are not dramatic animals. They are sturdy, whiskered catfish that spend much of their lives close to the bottom, feeling through murky water and sediment. That ordinary appearance made the lesions impossible to ignore. Anglers and wildlife biologists began reporting a striking increase in affected fish from Lake Memphremagog in 2012. By 2014, surveys suggested that nearly 1 in 3 brown bullheads carried the dark growths. The lesions were not all identical. Some were flat black patches. Others rose from the skin. In severe cases, melanoma spread beyond the surface into organs including the liver and brain. A cluster that large demands an explanation. Brown bullheads are often studied as indicators of environmental quality because they live near sediment, where contaminants can accumulate. So pollution was an obvious theory. A cancer-causing virus was another. Both ideas could explain why many fish in one region became sick. They could not, by themselves, explain what the researchers found when they examined the genomes. To understand that result, we have to separate two very different meanings of the word contagious. Some infections can raise the risk of cancer. A virus enters a body, disrupts cells, and may help one of those cells become malignant. If the infection reaches another animal, it can trigger a new cancer there. But the two tumors are not the same organism. Each one grows from the cells of its own host. A clonally transmissible cancer is different. It begins as an ordinary cancer in one founder animal. Then living cancer cells leave that animal, reach another member of the species, evade its defenses and continue growing. The infectious agent is not merely a virus carrying instructions. The infectious agent is the tumor cell itself. Stop on that idea for a moment. A cell that once belonged to one animal becomes a lineage that can outlive that animal. Every new host is not the parent of a new cancer. It is another habitat occupied by the same evolving clone. That claim sounds almost impossible. Fortunately, it leaves a genetic fingerprint. Imagine sequencing a conventional tumor from 2 unrelated fish. Most of the DNA in the first tumor should match the first fish. Most of the DNA in the second should match the second fish. Each cancer may contain mutations, but its basic genetic identity still points back to its own host. Now imagine that cells from one ancient tumor have been passing between fish. The tumor inside fish A and the tumor inside fish B should resemble each other, even when fish A and fish B have different genomes. The tumors should share mutations and variants inherited from a common cancer ancestor. That is the pattern the team reported in *Nature* on July 22, 2026. They used whole genome sequencing to compare tumor tissue with healthy tissue from affected brown bullheads. Tumors from different hosts shared hundreds of thousands of variants that were absent from the fish carrying them. The tumors formed their own genetic branch. Researchers also searched the analyzed tissues for a viral or microbial explanation. They did not find a viral or microbial genus associated with the tumours. That does not prove that microbes or environmental conditions played no role anywhere in the lineage history. It does mean the genomic evidence overwhelmingly supports the cancer cells themselves as the transmissible entity. The mystery was no longer whether the black lesions were connected. The mystery was how a living melanoma clone crossed the space between animals. Nature has produced this escape more than once, but only under exceptional conditions. Dogs carry a transmissible venereal tumor descended from a cancer that arose thousands of years ago. The lineage spreads through direct contact and is often survived by its hosts. Tasmanian devils face facial tumor disease. Biting can transfer living tumor cells, and the disease has devastated some populations. 2 distinct transmissible tumor lineages have arisen in devils. Several bivalves, including clams, cockles and mussels, carry leukemia-like cancers. In water, living cancer cells can move beyond direct body-to-body contact, and some lineages have even crossed between related species. Before the catfish study, naturally occurring transmissible cancers were recognized in those 3 broad animal groups: dogs, Tasmanian devils and bivalves. The brown bullhead melanoma became the 4th known type and the first documented in any fish. That does not mean it appeared recently. It means science recognized it recently. The route of transmission is still unknown. That sentence is essential, because the most interesting possibilities are not yet confirmed. The disease appears in larger fish of spawning age. During reproduction, brown bullheads crowd into shallow water and make close physical contact. Scratches, bites, or exposed tissue could create an entry point for living tumor cells. Another possibility is the lake bottom. Bullheads are scaleless, bottom-dwelling fish that spend their lives against sediment. If tumor cells can remain viable outside a host, sediment or water could help carry the lineage. Environmental stress may matter too. Researchers reported elevated arsenic in the cancer cells and noted a geographic relationship with naturally occurring arsenic in regional soils. Pollution, hormonal changes, or other stressors might influence immune defenses. But none of these ideas has yet been established as the root or the cause. The careful conclusion is narrower and stronger: the tumors share a clonal genome, and the living cancer cells are supported as the thing being transmitted. Everything between one host and the next remains an open investigation. The strangest clue may have been written before genetics existed. In July 1852, Henry David Thoreau described a catfish with what he called a very large velvet black spot. In 1858, he recorded another diseased fish with an inky black crust covering much of its head. The descriptions sound familiar. They do not prove that the modern cancer lineage was already moving through New England waters. There is no preserved genome linking those fish to the tumor sequence today. But the notes change the scale of the question. The lineage might be recent, or it might have been hidden in plain sight for generations. Researchers are now looking beyond Lake Memphremagog. Similar disease has been reported in other regional lakes and ponds. Establishing whether those tumors belong to the same clone could reveal how far the lineage has traveled and perhaps where it began. This story contains the words contagious cancer, so precision matters more than drama. The researchers report no known risk to humans. There is no evidence that this catfish melanoma can infect people, and the finding does not mean ordinary human cancer is contagious. Transmissible cancers survive only when a rare combination of cell biology, contact, environment, and immune escape allows living cells from one animal to establish themselves inside another. The ecological risk to the bullheads is less clear. Some affected fish survive with tumors for years. Researchers have not yet found the sharp age collapse that would reveal a rapidly fatal disease across the population. But tumors can invade internal organs, and the long-term effect on survival, reproduction, and abundance is unknown. That uncertainty is not a reason to look away. It is the reason to keep watching. The deepest twist is not that a fish can catch cancer in the everyday sense. It is that one cancer can acquire an existence measured across many fish. Its original host is gone. Yet descendants of that animal's mutated cells may still be alive, dividing inside bodies they did not come from. The lineage does not think. It does not plan. Natural selection requires neither intention nor awareness. But if cells that move more effectively reach new hosts, evade immunity, and survive, those traits can persist. The tumor begins to face evolutionary pressures that resemble the pressures on a parasite. That is why this discovery matters beyond one lake. It reveals how thin one of biology's boundaries can become. A body is normally the complete world of its cancer. In a few extraordinary lineages, that boundary breaks, and the disease becomes a traveler. How many transmissible cancers look like ordinary wildlife disease simply because nobody has compared the genomes yet? Tell us which hidden ecosystem scientists should investigate next. Subscribe to CreatureCosmos and join our community for real animals, real evidence, and discoveries that make Earth feel unknown again.